Hi guys, it's Mr. Pollock Biology here with my second video on DNA technology from Unit 5. In this video we're going to look at in vivo gene cloning and the use of plasmid vectors. So here's our objectives. We're going to describe the action of DNA ligase, we're going to understand how genes can be inserted into a plasmid vector, and we're going to explain how host cells containing the recombinant plasmid can be identified. So let's get started by looking at DNA ligase. So DNA ligase, its job is to join together DNA fragments that have complementary sticky ends. The thing is, sticky-ended fragments will automatically anneal together. They will stick back together, but the hydrogen bonds will only form between the complementary base pairs, which means that they can be broken pretty easily. So DNA ligase, its job is to complete the sugar phosphate backbone so we get a stable double strand of DNA. And this is really useful because it can be used to piece together many different DNA fragments. So if we pick off where we left off in the last video, here's two sticky-ended strands of DNA, so fragments, and these have been produced by cutting with the same restriction endonuclease. So the complementary base pairs are there, and that means we can use DNA ligase to form the proper double-stranded DNA by completing the sugar phosphate backbone. And this is recombinant DNA, where it's been formed from two different sources. So how do we get this into a host cell or a living host cell? Because at the end of the day, that's what in vivo means in living. So to get it into a living cell, we need to use a vector, and we use plasmids as vectors, which are circular rings of DNA that are found in some bacteria. I won't say most and I won't say all, but some. And here we have an example of a plasmid. So plasmids often contain the genes for antibiotic resistance. So I've got in blue the gene for antibiotic resistance to tetracycline. And in dark blue, I have the gene for antibiotic resistance to ampicillin. So if a bacterium was to contain this vector, was to contain this plasmid, it wouldn't be able to be killed by tetracycline or ampicillin. So if we want to insert a fragment into this plasmid to insert into a host cell, we would cut the plasmid and we would cut it pretty much smack bang in the middle of one of the antibiotic resistance genes. And we would cut it with the same endonuclease that we use to produce the DNA fragment that we wish to insert. So here we are, there's our DNA fragment, and we can use DNA ligase to form a recombinant plasmid. There we are. Now note at this point that the gene for antibiotic resistance to tetracycline has been disrupted, whereas the gene for ampicillin resistance remains intact. So that means that this bacterium, if it contained this plasmid, could be killed by tetracycline, but not by ampicillin. So we need to get this vector, this plasmid here, into a bacterium. And we do this by something called bacterial transformation. Basically, it's a case of taking your modified plasmid, your recombinant plasmid, taking your bacterium, adding calcium ions and heating, and that makes the bacteria permeable. So the plasmid will be taken in through the cell wall and the cell membrane. There's a problem with this though, because it doesn't always work. It's not massively successful, and in fact, very few bacteria, around 1% or less, will actually fully take up the plasmid. The other problem is that the recombinant plasmid doesn't always form because the sticky ends where the restriction endonuclease cuts the plasmid, those sticky ends anneal and stick back together before the DNA fragment is taken in. So we need to be able to confidently identify which bacteria have taken up the plasmid and which haven't. So how do we do this? Well, we need to understand a couple of things. So here's a bacterium that has no plasmid. This guy can be killed by antibiotics. This guy here has taken up the unsuccessful plasmid, the one that didn't take in the fragment. This can't be killed by tetracycline or ampicillin because the genes are still intact. This guy took up this plasmid successfully, and it's the right plasmid as well. This guy can only be killed by tetracycline. Ampicillin will not kill it because the gene for ampicillin resistance is still intact. And we can use a couple of different ways to identify successful hosts. The first of which is a really nice example of the old way of doing things, which is replica plating. Basically, you would grow a bunch of bacteria on some agar jelly. From there, you would take a copy, so you would basically just press on another agar plate on top of the original plate to take like a fingerprint of it, and you would grow it again. This time, though, it would be on a plate with ampicillin. So this would kill off all of the organisms, all of the bacteria that were not resistant to ampicillin. You would then do the same thing again, but with a tetracycline plate. Okay, so by doing this, you can see which bacteria have taken up the plasmid, and more importantly, by comparing the tetracycline plate to the ampicillin plate, you can see which organism have taken up the correct plasmid, or the one that's been made recombinant, because at the end of the day, the recombinant one will be able to be killed by ampicillin, but not by tetracycline. So it will be the colony that's present on the ampicillin plate, but not on the tetracycline plate. There are other ways of doing this, more modern techniques. For example, we can use fluorescent gene markers, which is basically where we insert the gene for a fluorescent protein, which is found in jellyfish called GFP, and then we insert the desired gene that we want in the middle of that GFP gene. And bacteria which do not show the green fluorescence have therefore taken up the plasmid successfully. We can also use enzyme markers to do this. So you could insert the gene for lactase, and basically lactase will turn a specific colour of substrate blue. The desired gene you're then going to insert right in the middle of that lactase gene. So bacteria which do not produce the lactase will not be able to turn the substrate blue, and can therefore be said to have taken up the plasmid. This is quite complicated. It might take a couple of watches to get your head around this, but do take your time with it. It can be tricky. So to summarise, a DNA ligase is used to form recombinant DNA. Plasmids cut with the same restriction endonucleases are used as vectors. Identifying host cells which have taken up the vector can be achieved by using gene markers for antibiotic resistance, fluorescence, and production of an enzyme. I hope that's been helpful for you guys. Thank you very much for watching. Please like, comment, and subscribe.